Bone contains 4 cell types: osteoblasts, osteocytes, osteoprogenitor cells, and osteoclasts. Osteocytes are mature bone cells. They maintain and monitor the protein and mineral content of the surrounding matrix. The minerals in the matrix are continually recycled. Each osteocyte directs the release of calcium from bone into blood and the deposition of calcium salts into the surrounding matrix. Osteocytes occupy small chambers called lacunae that are sandwiched between layers of calcified matrix. These matrix layers are called lamellae. Channels called canaliculi radiate through the matrix from one lacuna to another and toward free surfaces and adjacent blood vessels. The canaliculi connect adjacent lacunae and bring the processes of neighboring osteocytes into close contact. Tight junctions interconnect these processes and provide a route for the diffusion of nutrients and waste products from one osteocyte to another across gap junctions. Osteoblasts are cuboidal cells found in a single layer on the inner or outer surfaces of bone. These cells secrete the organic components of the bone matrix. This material, called osteoid, later becomes mineralized through a complicated multi-step mechanism. Osteoblasts are responsible for the production of new bone, a process called osteogenesis. It is thought that osteoblasts respond to a variety of different stimuli, including mechanical and hormonal, to initiate osteogenesis. If an osteoblast becomes surrounded by matrix, it differentiates into an osteocyte. Bone tissue also contains small numbers of stem cells termed osteoprogenitor cells. Osteoprogenitor cells differentiate from mesenchyme and are found in numerous locations, including the innermost layer of the periosteum and the endosteum lining of the medullary cavities. Osteoprogenitor cells divide to produce daughter cells that differentiate into osteoblasts. The ability to produce additional osteoblasts becomes extremely important after a bone is cracked or broken. Osteoclasts are large multinucleated cells found at sites where bone is being removed. Osteoclasts are derived from the same stem cells that produce monocytes and neutrophils. They secrete acids through a process involving exocytosis of lysosomes. The acids dissolve the bony matrix and release amino acids. The acids dissolve the bony matrix and release amino acids and the stored calcium and phosphate. This erosion process, called osteolysis, increases the calcium and phosphate concentrations in bodily fluids. Osteoclasts are always removing matrix and releasing minerals, and osteoblasts are always producing matrix that quickly binds minerals. The balance between these two activities is very important. When osteoclasts remove calcium salts faster than osteoblasts deposit them, bones become weaker. When osteoblasts are more active than osteoclasts, bones become stronger and more massive.